Thank you, and I appreciate the invitation to share some of our work today. Um, I think that most of you are aware that AAV vectors have been used uh, quite robustly both in preclinical and now clinical trials. There's a small number of approved products. And for an AAV vector, we basically remove the viral genes, rep and cap, and put in our therapeutic expression cassette or the DNA for which we want to do some editing with, as you heard about from Lee and previous speakers. But one of the major issues in using recombinant AAV vectors is the ability to predict from animal studies into human studies in terms of selecting the vector that will most replicate or most basically work well from studying these animal models. So, many people use rodents. There hasn't been good concordance between rodents and humans. There's NHPs, which again, there's not perfect concordance and much more difficult to use. There's a humanized mouse liver model for one that we've used. People have used human organoids, now even whole liver explants. And to really test it, if we were using back in biblical times, we could have used Noah's Ark and at least got an n=2 for each species. Now, what we did a number of years back is that we took an AAV capsid library, which is something we described back, several decades ago, where we have millions of different capsid variants. And we would inject this into a mouse that had a chimeric liver comprised of both mouse and human hepatocytes. And we would treat the mice with wild-type adenovirus, which allowed the AAVs that entered the human liver cells to replicate. We would take those out and we would isolate the AAVs, and then we would do recurrent screening or selection in these humanized livers. And at each stage, we would do full-length, high-throughput sequencing. And then we would take the sequences for the capsid themselves that were being overrepresented after the multiple passages in the animal model and vectorize these and test them. And the initial tests were done in the humanized mouse model. So we made a simple GFP expression vector. We'd inject these into the mice with the chimeric livers, and we would stain for look for GFP activity. And then we would also look and stain for a human marker, in this case, a human enzyme. And then we can merge and we can calculate the number of mouse cells and the number of human cells that are GFP positive. And we compared this to a vector of AAV8, which was kind of the gold standard. Now, AAV8 turns out to work extremely well in mouse liver, but actually isn't that great in the humanized cells in the mouse liver. And one of the chimeric capsids that we pulled out of these screens, and we've pulled out a number now, is called LKO3. And this one works really well in the human cells, but interestingly doesn't work well in mouse cells. And this is true of several other capsids or chimeric capsids that we've pulled out in multiple screens. Anyway, so the question is, why is the AAV not working in one species versus another? And one of the simple ideas, and I thought it would be trivial, was the fact that there would be different receptors between the species. But Shin, who was a postdoc in the lab, started these studies, and they were followed up by Adriana, and now there's a team of other folks in the lab who are doing various aspects of this work. But we found something very different. So it turns out that the AAV actually gets into the cells equally well. But okay, first let's start off in the left-hand side with panel A. We're comparing two different AAVs that express luciferase, LKO3, which is the chimeric, that works well in the humanized system. And I should point out is being used in 2 clinical trials right now, and DJ, which is another chimeric that seems to work well in both species. And if we look at the luciferase activity, this is a log scale, and we're seeing a 2 to 3 orders of magnitude difference in transgene expression, where DJ works equally well both in mouse and human. And if we look at the messenger RNA levels, they're also correlate with the protein level activity. But if you look at the copy number of genomes that get in the cell, they're actually pretty similar. And if you look at the nuclear copy number of the vector genomes, there's maybe a slight difference, but not much in terms of what gets in between mouse and human. It's maybe a 2-fold difference, but that can explain the 2 to 3 order of magnitude difference in transgene expression. And it's not a specific promoter or transgene, as we've found that the same type of results occur if we use different promoters or different transgenes. So the discrepancy is not related to entry in cultured cells, and we found the same kind of thing occurs in mouse liver in vivo. If we look at LKO3, the expression here is 3 orders of magnitude difference, but yet the amount of DNA that gets into the nucleus is maybe 2.5, at most 3 times difference. So so there's clearly a difference in expression, but not in the amount of the DNA that gets into the nucleus and becomes double-stranded. So the question is, what is left? So one of the things we decided to look at was the epigenetic state. And we used the cut and tag technology, which allows us then to look at the various chromatin proteins that are associated with the vector genome DNA within cells. So this is an example of an experiment where we looked at LKO3 and DJ in human, which is H, or mouse. The green histone marks are the histones that are associated with permissive transcription, and the red ones are associated with repressive transcription. And what we see is that in the human cells, in the mouse cells, there's a paucity of the active the histone modifications that are associated with active transcription relative to the human cells. The repressive marks are about equal concentration, both in human and mouse cells. So what we're seeing then is that based on the capsid that's delivering the AAV vector genome, there can be a difference in the epigenetic marks. So one of the things we did was we did alignments of different AAV capsids that we knew worked better or worse in one species versus another. And we looked at alignments, and we decided to add a single amino acid insertion in position 266 in the capsid sequence that would basically then push the alignment over to the right. We can do structural predictions, which the significance of we don't know. And we come up with a new capsid that's called AAV-AM. And this actually then takes an LKO3 capsid, adds 1 amino acid to the capsid sequence, but it restores the ability to see expression in the mouse cells compared to the human cells. And again, the expression of the protein activity correlates with the messenger RNA expression, and the AM copy numbers, again, can explain the differences in expression. But if we look at the permissive histone marks, what we find now is that AM, both in the mouse and the human, are highly concentrated compared to LKO3. So changing one capsid amino acid sequence now allows the vector genome to be associated with permissive histone marks and provide robust transgene expression in both species. And this is just showing you the data for 3 different capsids AM, LKO3, and DJ. And as I've told you, the expression appears to correlate with the concentration of the permissive histone marks. And this also occurs in vivo as well. If you look at LKO3 injected mice versus AM injected mice, you can see that there's almost no luciferase expression with AVLKO3, but with AM, we get very robust expression. And if you look here, you can actually measure the luciferase activity by taking out the liver and doing the enzymatic assays. And again, the protein activity and the enzyme activity correlates with the messenger RNA level, but which here in the first two panels are in log scale. But if you look at the vector genomes per nucleus, this is a linear scale, and the difference here is about 2.5 times difference, which again can explain the huge difference in transgene expression. So what this is telling us is that the capsid protein itself is somehow allowing the vector genomes to be set up epigenetically in the target cells. And changes in the capsid sequence can alter the epigenetic marks that occur on the vector genome. So one of the questions we wanted to ask is, if you use a capsid that actually is restricted in in mouse cells, and then co-inject it with a capsid that is permissive in the mouse cells, can you trans-complement the activity? And the answer to that is no. So the capsid effect of how it sets up the epigenome occurs in cis and cannot be complemented in trans. So our current model is that the LKO3 capsid, when it enters a mouse cell, the capsid uncoats in the nucleus, but it sets up a chromatin structure that's unfavorable for transcription. Whereas in a human cell or a mouse cell, the AM capsid, which varies by 1 amino acid, is now being set up in a histone favorable structure that allows for transcription to occur. Now, another interesting thing is that if you look in the clinical trials that have been published, there's a very wide dose response between individuals, and the reason for this isn't known. And we actually recently did a non-human primate comparison where we were comparing two different capsids. We compared LKO3 with a new capsid that we developed in our lab, KP1. And we used a human factor IX as the expression protein, which is a secreted protein. Now, it's well known that non-human primates will develop inhibitors when you express human factor IX, and you do get loss of expression over time. But interestingly, there was one animal in each group that didn't express well from the beginning before the antibodies were formed. And if you look at the DNA copy number, or the vector genomes per diploid genome, within each group. Now, we know LKO3 now works better than KP1 and gets into the cells better based on DNA copy number. But if you look at the DNA copy number between the individual animals, there's really very little difference, both in the LKO3 and in the KP1 group. But if you look at the transcripts, again, the transcripts correlate with the protein expression early on. And if you then look at the histone marks in these animals, you see that the animals that didn't express well from the onset actually have a reduced number of the permissive histone marks. And another interesting thing is, both in humans and non-human primates, we've never been able to get to the same level of expression that one can get in a rodent with the best capsids that are being used. So when we look at our histone marks, we actually correct for genome copy number. And if you look at the permissive histone marks between mice and the non-human primates, we find there's a 35 times higher level concentration of the permissive histone marks in mouse injected in mouse liver than we would see in a non-human primate liver. So this suggests that there are inherent differences between rodents, let's say, and primates in how they set up their epigenomes. So just to summarize, the AAV capsid during the encoding in the nucleus is participating in how the epigenomic state of the vector genome forms. And this is a functional end biomarker. We're very interested in understanding the molecular events that occur to allow this to epigenetic state to be set up. The variations in the capsid sequence can influence the epigenome and hence transgene expression. And again, I think importantly, there may be differences in how AAV vector genomes are epigenetically marked in lower, if you will, versus higher mammals. And the variation in expression between non-human primates and humans may be explained by different histone modifications based on polymorphic variants, let's say, in other genes that may affect chromatin modification. And besides our own non-human primate data, there's also some human data that may suggest this as well. So I think that in order to understand why people have such a wide dose variation when they're injected with an AAV vector may be explained if we can understand these molecular events. And the reason that this is important is if we understand these molecular events, perhaps they would be druggable targets that could enhance transgene expression, or at least allow us to predict what dose to give in an individual human as we move forward. So I've mentioned the people in the lab that did most of this work, and I want to thank the other members of the lab as well, and I'm happy to answer any questions. Thank you.